So I'm going to hand you over to our speaker for this evening. The topic, I think, needs no introduction. Look vaguely around you. But our speaker is Liam Brierley. He's an epidemiologist and research fellow at the University of Liverpool and a media ambassador for the Royal Statistical Society. His research interests revolve around using data to understand why viruses jump between host species and to predict future outbreaks of infectious diseases. And here to tell us what is going on with this coronavirus COVID-19 stuff, please welcome your speaker for the evening, Liam Brierley.
Happy also if people want to put in some clap emojis in the chat there. So yeah, as Marsh said, I'm Liam Brierley. I'm a research fellow at the University of Liverpool. I'm an epidemiologist interested in the ecology, the evolution, and the genetics of emerging virus outbreaks. So I'm obviously very interested in our current pandemic. And tonight I'm going to talk a bit about where it came from, but also the infodemic. Now my Twitter handle is there, it's L_Brierley. If you can't think of a question right now, but you think of one later, I'm totally happy to field questions through Twitter after this evening's session. So in the next sort of 20-25 minutes, what I'm going to do is I'm going to put the emergence and the outbreak of COVID-19 into the context of what's come before and what to expect in future. I'm going to review evidence for the origins of the novel coronavirus in a nutshell. And I'll also talk about how some flawed research has led to the spread of misinformation about the epidemic's origin. Now, I also think it's really useful for me to explicitly say here now what I am not going to do as part of this talk. So I'm not going to be discussing transmission and spread in this country or anywhere else, because what we know epidemiologically changes so rapidly, I can barely keep up. This is also not a talk about infection control or risks or symptoms, the latter because simply I'm not a clinician, and the former is because I can't really say anything that hasn't already been said. So it probably doesn't actually need an introduction, as Marsh says, but to reintroduce our current topic, I wanted to start with maybe a quick lesson in names. And that's not just because I take unreasonable joy in needless pedantry, but it's because there's been a lot of confusion about the names of the virus. So we talk about coronavirus, but the word coronavirus really just refers to any of the viruses within the family group Coronaviridae. So this includes other epidemic coronaviruses, but it also includes some coronaviruses that cause a common cold, and it also includes many that don't actually infect humans. Now, coronaviruses is the plural of coronavirus, and the new virus that emerged in Wuhan in December is properly named SARS-CoV-2, whereas the disease caused by this new virus is named COVID-19. So if you're asking, will I catch coronavirus? It's a little bit like asking, do you own mammal, if you're trying to figure out if somebody's got a pet cat or a dog. And why are these names so different? Well, it's because viruses are named by the International Committee for the Taxonomy of Viruses, and diseases are named by the WHO, and they each operate by their own kind of different rules. And some have argued this doesn't really make sense, but for now, this is what we have to work with. And if we look at the current situation in terms of global case estimates, as of today, it looks like this. There's about 487,000 cases of COVID-19 worldwide, and these are just the confirmed cases, and these are in 175 countries, and we are still in an exponential rise of cases at the moment. So where do new viruses like SARS-CoV-2 actually come from? Well, the majority of human viruses, about 60-70%, are what we call zoonotic. And this means that they can be transmitted from non-human animals to humans. It's often animals that we have a lot of contact with, like domestic animals, or it's animals that we're closely related to, like primates, but it also includes many other wild mammals and birds. And SARS-CoV-2 is certainly not going to be the last brand new human virus. It seems very likely that we're going to continue to see outbreaks of previously unknown viruses, because zoonotic transmission is constantly happening, such that we recognize about 2 new human viruses each year on average. And that's been surprisingly constant since the 1940s, 1950s. Our most recent estimate was that there are 214 human virus species. So to move on to the epidemic coronaviruses, many people likely remember that SARS coronavirus emerged in China in 2003, caused about 8,000 cases, had a case fatality ratio, or CFR, of about 10%. And then later, MERS coronavirus emerged in the Middle East in 2012. This has caused about 2,500 cases to date. It still occasionally causes infections. We see the odd small outbreak starting of MERS. The case fatality rate of MERS is higher at about 30 to 40%. Now, in this image, what I've done is I've scaled the area of these circles to be proportional to the numbers of cases and numbers of deaths. So if we move on to the novel SARS-CoV-2, it's caused many, many more cases than previous epidemic coronaviruses. It certainly seems like we're going to have over 500,000 cases by tomorrow at this rate of increase. Now, it's really important to note that these vary in risk. So while MERS-CoV is the most deadly, it's the one that you'd least want to have, it's the one with the most individual risk, SARS-CoV-2 has presented the biggest population risk. Even though the fatality rate is much lower, we've still seen an overall higher death count. This red circle is still bigger. Now, SARS-CoV originated in bats, but evidence has shown that it actually first transmitted to the masked palm civet, and this might be the cutest baby animal that I've ever seen in my entire life. From there, it transmitted to humans through contact in markets in Guangdong. The MERS-CoV originated in bats as well, and then it transmitted to camels, where evidence suggests it circulated in camels for a while before jumping to humans in Saudi Arabia. Now, the question now under investigation is, where did the current pandemic come from? How did SARS-CoV-2 reach humans? Well, to answer that question, we need a modern science known as bioinformatics. Now, what is bioinformatics? Now, your first guess might be one of the friends of a particular comic book Gaul, but bioinformatics is broadly defined as the science of processing, extracting, and understanding information from complex biological data. I'm going to narrow this down a little bit. I'm going to narrow this down here to mean the science of extracting biological relationships from complex genetic sequence data. Essentially, the genetic code of all organisms is made up of one long, big string of coded information, and viruses are no exception to that. And we can use that information to tell us things like evolutionary relationships. So, for example, here I've got some genetic code from 3 viruses, and it's a little bit like a visual sort of matching puzzle. If you just sort of skim your eyes over these sequences for a moment, hopefully you'll have already spotted that the top and middle virus are actually very similar, whereas the bottom is quite different. So this already tells us something quite important. It tells us that the top and middle are more closely related, and so one might have evolved from the other, or they might share a common ancestor, whereas the bottom virus looks quite unrelated. So we can apply these sort of principles and compare SARS-CoV-2 to viruses that we already know about in different hosts to try and find out which is most similar. And by doing that, we might get a clue as to where the virus has come from. Bioinformatic analyses have shown SARS-CoV-2 to have 96% genetic similarity to a bat coronavirus from a horseshoe bat like this one. So it seems very plausible this new virus originates in bats, just like the other epidemic coronaviruses. However, one of the areas that has the most differences between SARS-CoV-2 and this bat coronavirus is this part. It's a protein found on the outside of the virus, and instead, this looks very, very similar to the same part on the outside of a coronavirus that we found in a diseased Malayan pangolin. So here's this protein sequence comparison along the bottom there, pointing out the areas unique to SARS-CoV-2 and the pangolin virus, but not the bat virus. So it's exactly almost like that visual puzzle that I just showed before. Now, there has been second-hand reports that pangolins were kept around this seafood market where the outbreak started in Wuhan, but that still isn't really enough to say whether the virus has gone from bats to pangolins to humans without more investigation. There could yet be any number of other animal hosts involved. Now, for the original SARS outbreak, these investigations took basically the best part of a year. But we already knew this for SARS-CoV-2 in mid-February, because genetic sequencing of the virus was done super quickly within just 2 weeks. Now, kind of parallel to that, we've also seen a rise in the speed of open science and preprint sharing in the last couple of years. If you're not familiar with what a preprint is, ordinarily a scientific paper has to undergo this peer review from experts in the field before it's actually published and it becomes available to read. But many scientists now are choosing to make their work available before or during this review. And this has now become actually quite widely ingrained in academic culture. So here we've got the rate of preprint posting for a couple of previous outbreaks, including Ebola there in green in 2004, right through to just seasonal flu starting last year in yellow. And gradually, we've seen more and more use of preprints to share knowledge as an epidemic happens. I've just put a little notice there, by the way. All this data is open, and I've made the code to create all these plots available. So that's on my GitHub, which I can also link after this talk. So those are some previous epidemics. But if we look at the rate of preprints for SARS-CoV-2, we can see that research posted about the current epidemic started super early, just 22 days after the first reported case cluster. We had some preliminary research reports, and it's absolutely boomed since then to this kind of scale that we've never seen before. We've had almost 800 preprints in the first 3 months, and we've never had this kind of flood of information during an active epidemic before. Now, while the large number of researchers posting their research super early has led to some really important knowledge sharing, having all this unreviewed research openly available has been a bit of a double-edged sword in a way that perhaps we weren't actually prepared for. What I mean by that is that we've seen some unreviewed preprints get lots of attention that actually had unreliable findings. So one preprint that claimed to have found genetic similarity that indicated material from HIV had either naturally or artificially made its way into SARS-CoV-2. Now, the problem with this claim is that the segments of genetic code they looked at were very, very short. The largest segment was only about 0.08% of the genome. And that's so short, their information is really actually not specific to HIV. And the exact same code can be found in lots and lots of other organisms just by random chance, including that closely related bat coronavirus. So this actually doesn't give us any evidence whatsoever that HIV has contributed to SARS-CoV-2. And the best way I can describe it is I've got a tweet from biology teacher Jeff Witters here, because I can't think of a better sort of analogy for it. To have such a level of confidence from this to describe finding the same super short sequence in both HIV and SARS-CoV-2, to be describing that as uncanny is a bit like finding a short match of words in a rambling manifesto posted by a serial killer to a random line in a Harry Potter book and then arresting J.K. Rowling on that basis alone. So this paper was highly criticized and it was withdrawn 3 days later. Now, you might have also heard that snakes are the origin of SARS-CoV-2. Now, this came from an unreviewed analysis in the Journal of Medical Virology. And what these authors did was they essentially looked at the virus and the closely related bat coronavirus, and they looked at how often certain patterns appear within their genetic code, and they compared that to many types of animal. And this is the result. So essentially, the animals are the columns here, and whether it's red or green doesn't really matter. just the more similar the colours are across these columns, the more similar the pattern usage. And they looked at this and they concluded snakes were the host of SARS-CoV-2. Now I wonder if people can see what the problem is. Well, there's another bit of a clue in this, this tree at the top. But really, if you're looking at this figure, these patterns are actually showing SARS-CoV-2 to be really strongly similar to the bat coronavirus, certainly more similar than it is to snakes. So why would you conclude snakes are the host and not bats? Now, quite a few virologists came forward to discredit this based on the fact this particular flavour of analyses is actually never used to determine what kind of host the virus comes from. And quite a few epidemiologists, me included, came forward to discredit this based on the fact that humans share almost none of our RNA viruses with reptiles. That's not to say we won't ever see that in the future, but that alone should have put big question marks on this from the start. And in fact, several works came out after to formally repeat both of these preprints showing their flaws. So in both of these, it seems like biological plausibility has taken a bit of a back seat to immediacy in producing results. Bioinformatics is a super powerful tool, but at the core of it, that's all it is. It's just a tool. It has to be used in the right way. And that takes human critical thinking. And there's no advanced method, there's no AI or anything like that that is ever going to take away that step. The problems are caused by over-reliance on trusting outputs and methods without really dissecting what they're doing. Now, even though other scientists were actually quite quick to discredit these works, because they were immediately publicly available, their message had already gotten out there. And the WHO has quite appropriately referred to this, this uncontrolled spread of misinformation about COVID-19 as an infodemic. And it's tempting to hear that word infodemic and think that it only ever originates with social media or maybe print media. But considering the role of research, I would argue the infodemic is more systemic. If you look at Google Trends, which are measures that Google produce on popularity of certain search terms, the posting of these preprint articles was followed by an immediate rise in associated misinformed searches. So searches for HIV and coronavirus, they're in blue, and snake and coronavirus in green. Now, obviously, I can't be sure what the intent of these searches were. Some of this might have been fact-checking, for example. But what I'm trying to show is that these unsound findings did quite quickly filter through into public perceptions. And I've added this just last moment, actually, as the results from a particular survey are now available. This survey was done in late February, so basically just at the end of that previous graph of search terms. And one of the questions in this survey asked whether it's likely the new coronavirus is a manufactured bioweapon, which is how the results of this HIV preprint were interpreted in some media sources. And among 3,000 US and 3,000 UK respondents, just under a quarter said it was slightly likely or stronger in the US, and just under a fifth in the UK. And even that level of belief, I think, is actually quite staggering. Now, another piece of misinformation that I've added quite last minute is this. There was also a press article claiming that there was a link between the SARS-CoV-2 outbreak and bat consumption in soup. And this included a couple of what you might call shock images, despite these images not actually being taken in China, and despite there being no scientific basis for transmission via food consumption. Now, this was later revised to remove the link between soup and the outbreak, but immediately after publishing, we saw this spike in searches about coronavirus and soup. And I've included this just to show the level of misinformation related searches that came from this preprint research was basically just as high as the level of misinformation related searches that came from mainstream press media. So if unreviewed research is now seeding in actually a significant part of that infodemic, what can we actually do about it? One of these preprint platforms, one of these websites that hosts them, responded by putting up a site-wide disclaimer that reiterated all the research there is not peer-reviewed and it shouldn't be regarded as conclusive or used to guide clinical practice and so on. Leading scientists have also made public announcements and signed statements against conspiracy theories. But I think the core question is, if you consider now this huge amount of information that's out there in unreviewed research servers, whose responsibility is it to regulate all of this? Is it the responsibility of scientists, even though they already contribute quite a significant workload in doing the peer review in the first place? Is it the preprint servers, maybe through volunteer screening? And some of them do this, although nobody can be enough of an expert in all fields. Or is it even the end reader? Is it the responsibility of the public and journalists and the media to actually know the limits of what they're reading? And it's probably, the answer is probably quite boringly all three. Although I think this has shown that we do need some serious guidelines now as to how the general public should approach preprints. To people outside academia, this distinction between peer-reviewed papers and unreviewed research is probably not very clear. So this is something that I'm trying to write a skeleton of myself at the moment, to try and create some guidelines as to how the public can actually read preprint research in a more kind of balanced way and understand what the limits are. So I'm going to wrap up. Genetic evidence suggests that, like the other epidemic coronaviruses, SARS-CoV-2 originated in bats. But we need more work before we actually know whether other animals are involved for sure. We've had this gap between academics and the public that's been gradually narrowing as the use of these open access preprints has increased and increased, and as a potentially unexpected result, unreviewed research can be a source of misinformation during epidemics. And I would argue that we need to be better prepared to combat this, not just during epidemics, but also during future large global crises. We can do this through good practice and transparency about preprint limitations. So I might usually sort of finish up by including some sort of talk acknowledgements. Instead here, I wanted to acknowledge something that you can do by just making everybody aware. If you're not already aware of it, there is a platform that is trying to crowdsource volunteer time to help COVID-19 research. So whether you have practical skills, data analysis skills, coding skills, or even none of them. Now, I'm not affiliated with this officially, I should say, but there's more info if you are interested at crowdfightcovid19.org. Thank you for joining me tonight.